Well, during these years, thanks to the support of the Italian Cystic Fibrosis Foundation, we found that CFTR interactome is not only composed by proteins, but also by lipids. And we found that the ganglioside GM1 is one key lipid involved in the regulation of the membrane stability of the channel, and the cholesterol, molecules that all of you know, is involved in the control of the motility of the channel belonging to the membrane and to reduce the motility of the channel, and this reduction is allowed to take interaction with the CFTR scaffolding protein and promote the function of the channel. Actually, we know that in bronchial epithelial cells where we don't have CFTR, where CFTR is absent, such as for the mutation of 508 deletion, what happened? The lack of CFTR in the cell plasma membrane is accompanied by a decrease in the content of the ganglioside GM1 and of the cholesterol. And what we found was that if we administered, for instance, GM1 in order to recover the plasma membrane level of this ganglioside, well, we demonstrated that this condition allowed to increase the effectiveness of the modulators that are the modulators of present in Orkambi formulation that are able to increase the rescue of the mutated CFTR, the CFTR with the mutation F508 deletion. So based on this evidence, we're wondering whether the exogenous administration of GM1 could in some way ameliorate also the effect of the ETI formulation. We administer ETI together with the GM1 in CFB cells expressing F508 deleted CFTR, and what we found, as you can see here, is a slight but significant increase in the quantity of the protein at the plasma membrane level. Actually, what we know from for other proteins is that the acyl chain of the ganglioside play an important role in the regulation of the stability of the channel. So thanks to the chemistry lab of in our department, we have the opportunity to synthesize GM1 analog with long chain fatty acid, not so long, but quite long chain fatty acids, and with 22 carbon, shortly C22, shorter one with 10 carbon atom, shortly C10, and more short with 4 carbon atoms, C4. And as you can see here, we found that the combined treatment with of ETI with the different analogs, in particular with the short chain gangliosides, showed a more recovery of the protein at the plasma membrane level. So, wonderful, no? We try also, thanks to the Servizio Culture Primarie, to check about the activity of the channel. But unfortunately, we were not able to find a more recovery than ATI in the treatment when we treated the cells with short chain GM1. We did the same with the cholesterol, but we selected to deliver cholesterol to CFB cells overexpressing F508 deleted CFTR in a more physiological way that we know, that is in the form of LDL. That, as you know, is the way in which cholesterol is transported in our body in all the tissue, in all the cells. And as you can see here, we found again that LDL together with ATI is able to increase the quantity of the rescued 508 deleted CFTR at the cell plasma membrane level, and fortunately, this time this result was also confirmed in primary culture by Servizio Culture Primarie. So we move on, and we try to study the possible effect of this lipid, possible adjuvant effect of this lipid in other CFTR mutation that affect the folding and the stability of the channel. We have some trouble because we think about the possibility to take this cell line from the North American Cystic Fibrosis Society, but these cells are not still available. So thanks to the Pedemonte's laboratory, we have the possibility to get the vectors coding for these isoforms, and we started to perform a transient expression of this vector in CFB41 or minus cells, and also is more or less 1 year and a half that we try to obtain cell lines stably overexpressing this isoform. The result that we obtained on transient expression was that GM1 together with ATI showed an effect on the variants N1303K and L1065P, and for what concerns cholesterol in the form of LDL, we found an effect in both of these two variants, but also in the variant W128-2R. Actually, this is the story related to the effect of the ganglioside, of the cholesterol, in general of the lipid, in the stabilization of proteins belonging to the cell plasma membrane. But what we know is that the GM1 is characterized also by an important effect in terms of in the immune system, acting on different cells in different pathway. And so since the immune response is in some way, is impaired in CF, we're wondering whether the administration of these gangliosides means restoring the plasma membrane level of these gangliosides have a beneficial effect. And thanks to Rimessi laboratory, we found that the administration of the GM1 is able to recover the xenophagic activity of bronchial epithelial cells. Means in CF, when we have the lack of CFTR, the xenophagic activity is impaired, so these cells are not able to clear Pseudomonas aeruginosa. If we administer GM1, this activity is restored. And we try also what happened with ATI. Also ATI is able to recover partially the xenophagic activity of these cells, but the major effect remains those exerted by the GM1. We check also at the inflammatory level, checking the expression of in cytokines involved in the inflammatory response. This was performed in Verona's lab, and we found that ATI slightly increased the expression of pro-inflammatory cytokines such as TNF-alpha, interleukin-6, interleukin-8, and when we administered also GM1 together with ATI, for what concerns interleukin-8, we have a slight reduction in the expression. We move on, and thanks to the help of CF-ACOR, to study the effect of the GM1 in the in vivo model represented by mice chronically infected with Pseudomonas aeruginosa. We start from wild-type mice, and we plan to extend in the next weeks the experiment also on CF mice. And what we found is that at the concentration of 1 milligram per kilogram, we have a reduction in the body weight loss, means these mice stay well, and also we have an increase in the clearance rate for Pseudomonas aeruginosa. We observe a reduction in the bacterial loading in the lung, and also a reduction in the number of neutrophils in the BALF. In conclusion, what we found is that restoring the level of GM1 promotes in some way the increase in the protein levels of different variants of CFTR affecting the protein stability and folding, and we observed an enhanced xenophagic clearance activity and reduced Pseudomonas aeruginosa invasion capacity in CFB508 del cells. The in vivo administration of GM1 in a mouse model chronically infected with Pseudomonas aeruginosa resulted in a reduced body weight loss, improved clearance rate, lower bacterial burden in the lungs, and reduced neutrophils count. For what concerns the cholesterol, we found that restoring the cholesterol levels, have an effect also in terms of activity, have an adjuvant effect also in the activity for LTI, for the mutation F508 deletion, and also for other variants that, again, are involved in the perturbation of the folding and the channel stability. Last but not least, I wish to thank all the persons that collaborate in this work, of course, the Italian Cystic Fibrosis Research Foundation for supporting our work, and all of you for your attention.